I felt so selfish, but I was very grateful that they had a good lead and we could leave and go do their thing and still get to watch the Bengals have some success. So we are still everyone's still coming in here, it looks like a little bit late, a couple minutes late, but that's okay. We're gonna start and go ahead and take a peek at what this week holds. Again, we have our instructional points of biology 3 times a week, right? Monday, Tuesday, Wednesday. We're going to do teaching lessons. And then Thursday at this time, I have a help session. I'm going to actually close my help session from earlier. And then I also hold those help sessions from 8 to 8:30 pre-class for those of you who need help. So don't forget that that's there. I don't have a lot of students use that opportunity, but it is incredibly beneficial. Awesome. Hey, Day. Awesome. So yes, please don't forget about those learning opportunities.
I also noticed that Mrs.
the-
Oh, sorry, I didn't mean to interrupt, but
No, you're
Mrs.
okay.
M is in here. You know, she's the one who does the MAP test contest results. She's in here
Yes.
today.
Yes, she is one of our assistant principals for the high school. We're glad to have her. So let's do our before we do today's goals. It's a Monday. Let's wake up our brains. Okay, I always kind of like doing this anyway, but especially on Mondays. Let's get ourselves a blank board. What did we talk about last week? Okay. Oh, good. We're already throwing out words. I know sometimes it's like, all right, I'm in biology. It is Monday. Mutations. Good. And mutations are changes in DNA. Or what else do we use? What is that other M word? Mistakes. Good. So mutations are mistakes in our DNA and RNA. Now, I don't want to do anything more to this board. You guys are blowing up chat, and I love that. What I want you to do now is I want you to put it on the board for me. I created a new link for today, so
I don't think we have tools.
Yep, I was just going to go check because I know this is a new. All right, there you go. You have permission to draw on the whiteboard. I want you to doodle up this board. Tell me what you know. Put what you're putting in chat. Put that on the board for me. I'm not sure, Pip. I'll have to check on that for you. Insertion, deletion, substitution. Good. Those are the types of mutations. Changes in our
Yeah, what I
DNA
was
and
saying
RNA.
is that about the thing, I remember there was like a thing, a drawing where if you nominate people for 7 mindsets, I don't think we got the winners of it yet, but I'm just asking Mrs. M about it.
Okay. Oh, I like this over here. Mutations are not always harmful, right? They happen. They happen all the time. So you don't have to freak out and be like, oh my word, maybe my cell's mutating today. All right, you've got mutations, right? Good. And some of them have beneficial effects. They can be harmful. Ooh, good. I like that our ultraviolet radiation, right? Our drugs, x-rays, smoking, those are things that cause mutations. What else can cause mutations? Oh, here it is. A result of copying. Right? We talked about how our cells are constantly copying. You guys are doing excellent. You're remembering most of the things from last week. Good. I saw cystic fibrosis is an example of deletion. What is one of those other mutation? Yes, causing diseases, sickle cell, cancer. Okay, the very last thing from last week that we talked about was, can mutations be passed on from me to my babies? Can mutations be passed on? A lot of people answering yes in chat. I would say it's yes-ish. How do we know when they get passed on? Ooh, super close, Dustin. Good. Not mitosis, but the cells that go through meiosis, good, are reproductive cells, right? So when we have a mutation that happens in our reproductive cells, that's when that mutation can be passed on. Okay. And for those of us who have mutations in all of our other body cells, those don't get passed on, right? Those are going to stay in my body. So yes, they can be passed on, but they have to be in a certain type of cell. I am super pumped because that was really, really, really a great review, and you covered almost everything we talked about last week. So I'm super proud of you. I didn't know if you could bring that all back on a Monday. I mean, that can be hard. Sometimes Mondays it's hard to get our brains going. So what we're going to do is take that information from last week and move on today. All right, and we're going to look at our genes and how it reads the DNA, how it reads the genetic code, and how the cells in different parts of our body are reading our DNA when there's so much to read. Okay, let's pretend you have a book that is really, really thick. That would be like your DNA. Okay, and your goal for your job, your role for your job is in that book. But do you need to read the whole book to understand where your role is? The answer is no, right? You don't need to read that whole book. Now, there are some books in that you need to read in entirety, but I should maybe say more of like an instruction manual, all right? And let's say it's like a Lego instruction manual, and it's got all kinds of projects, like every kind of Lego project that there's possibly to build in there, all right? And you're only building one project. Your job, your role is assigned to complete this one project. You don't need to read them all. I am not telling you that. That is up for you to decide. All right. So that kind of gives you a preview of where we're going today with what we started with from last week. So before we go, though, let's read our goals. Who would like to read today? We're going to read our goals. And for our new students that are still getting used to all of our daily procedures here, I would like you to tell me, Pip, and then I'll have Ireland go second, why we do the different colors of goals in our classroom, why we do
All
them.
right.
And then Pip,
So,
will you read the first two, and Ireland will read the second two goals for today?
sure. So basically, first things first, we all know this is known as the triangle of knowledge. Basically, the purple, as we know are on right now, is like the knowledge. And as we get higher and higher, we understand a bit more. And when we reach the blue evaluation, we eventually learn to the point where we finally, recognize it very well and we get to know it more. And, these could be on your unit tests and possibly your final, but I don't want to get it too ahead of myself, stress you all out. So let's just read the goals. So what is the lac operon? Something I never heard of. And when is the lac operon turned off? Turned on, I meant. Thank you.
When is the lac operon turned off? What causes the changes from infant to adult?
Perfect. Great job, ladies. Thank you. So, yes, so as we learn this information, we're able to do more things with it, right? So, up here, these are going to be projects. This is going to be teaching your peers. This is going to be able to take things apart and teach them from the back to the front or front to the back. The more familiar you get with these things, the more you're able to do with them. All right, that's what Pip was telling you about this triangle, and that's what we're going to do. So today we're down here at the base. All right, we're getting some base knowledge. So today is the fundamentals. It's the bones of this. And then as we work through the week, we're going to put some meat on the bones, and we're going to be able to work through this content. Okay, perfect. So let's take a peek at what is the lac operon. Pip said, I have never heard of the lac operon, and that's okay because you know what? I had never heard of the lac operon either. But we're going to use this lac operon as an example. All right, this is an example of how our genes are expressed. All right, what do you think of when you think of expressed? And let me just do this for a minute because I get real stressed out by all the content. Let's take this in pieces. What's the first thing that comes to your mind? It can be reasonable or just crazy when I say expression. You don't even have to say gene. We're not even getting as fancy as gene. If I say expression, what would you say? Don't even go sciencey on me. I'm going to just turn your eyes off. And now all of a sudden you're not in biology class, okay? This does not need to be, ooh, I like that, Aria. Facial expressions. Faces, funny faces. You think of a hand, okay? Yeah, good, because like when you talk with your hand, you can be have expression. Good. What else? Expression. What do you think about when I say expression? Sign language? Your personality, expressing who you are. I like that. Your eyes. That's good, Justin. Can't you tell so much about somebody's eyes? Like you can tell when my girls aren't doing what they're supposed to be doing, right? Behind the scenes. And I look at them and I'm like, it's the expression in my eyes. I don't have to say it. Good. Yeah, your raised eyebrows, mannerisms. Good. Okay. I love all of these things because this is what we need. This is what we need today is we're going to build upon what you know, and then we're going to incorporate that with a lac operon. All right. Because nobody has an idea what a lac operon is. So an expression is what? We talked about personalities. We talked about reacting with our faces. I like that. I like the reaction with our faces because lots of things happen through our day, right? Think of something that happened in your weekend that you reacted to. Maybe the snow. Maybe the snow. What was your expression to the snow? Oh, you're too thick, Jesse. Let's see this. Did you see my expression? Right. Sad, pain. What were your expressions to the snow? My youngest daughter was like, I'm freaking out. Freaking out. That's what she said when she got out of bed.
I'm not gonna lie, my mom was like so excited because now she has the opportunity to just sleep in. In other words, her and my twin brother now have a snow day.
That is fun,
And I'm
right? So
absolutely
that's
jealous.
exciting. I bet there are lots of fun expressions when people have snow days.
I'm absolutely jealous of my parents and my brother who have snow days, but not us.
I understand that. I understand that. There are lots of benefits to online school, but snow days are just not one of them. Good, excited about the snow. All right, so think about those expressions. Those expressions are triggered by something, right? They are regulated by something. There's something that happens that causes that physical expression in our face to be shown or heard in our voice. All right. And that is very similar to how each and every single one of our cells are kind of behaving. All right. So gene expression and regulation are good examples of this. So before I do all the talking and you guys start tuning me out, let's have Aria, do you want to read today? Ireland read, Pip read. Would you want to read, Aria?
It's for some reason it's usually me, Ireland, but I think Aria might.
Or would
If
somebody new like to read too?
anyone new, anyone new?
Oh yeah, your voice isn't feeling well today, is it, Aria? I'm sorry.
We haven't had anyone new for a while, but anyone new?
We don't have
I
anyone new to
remember
read. We love when you pop on your mic.
I remember in my geometry class that Caden read a slide once. He did good. Anyone? Don't, like Mrs. Jackson
No
once
pressure.
said,
We just
don't
don't
be
want to.
shy.
Nope.
What
Okay,
Miss
that's
Jackson?
perfect because Pip loves to read. How much do you want to read? You know what, Kai? If you read, you can read one sentence, one paragraph, however much you want. You want to go for it, Kai? You can read all of it, some of it, whatever you want. You can read a box of it. You want to read here? And we will not. 2 sentences. Go for it. I'm happy with that. And this is a prokaryotic gene. I know some of our words are weird. Lactose intolerant.
Okay. Used as an example of a prokaryotic gene expression, what happens when someone is lactose intolerant? They don't create the EMS lactase, so their bodies can't break down the lactose in dairy products.
That was so good. Thank you so much. I love hearing your voices. Seriously, that was amazing. I appreciate that. Way to get out of the box today on a Monday. Anybody else want to finish this up? Has Kai inspired somebody else to jump on and read about our E. coli?
Don't be shy. Don't be shy, like Miss Jackson once said. I do miss those days with Mrs. Witalik, though. She was a good science teacher.
All right, go ahead and read it for us, Pip. So E. coli.
All right. Great work, by the way, Kai. Welcome to the reading rotation. So, E. coli, prokaryotic bacteria that live in our large intestines, try to break the lactose down. How do they know when to get to work creating enzymes to break lactose down when lactose is present? Thank you.
Good job. Good job. So we're going to look at an example of gene expression with being lactose intolerant. First of all, is anyone lactose intolerant? I guess you don't have to share if you don't want to. I am not. I feel like I would be kind of sad if I was.
My mom,
my expression
I
would be
think my
sad.
mom is, because my mom, every time she drinks milk, it's just messes up her. But when she eats cheese, it doesn't. I don't know if she is, but I think it depends on the amount of it that's in it. But
Sure.
only when she drinks milk.
And a lot of people they connect lactose intolerant to just milk, but think about all the things that are dairy. A lot of us like cheese, right? A lot of us like, I don't know, ice cream. Ice cream is made of cheese. Oh, sorry, my mic went off. So, yes. So there are lots of things other than milk that might be a sad thing. All right. So, what happens when someone is lactose intolerant? Well, they don't create an enzyme, and an enzyme is one of those 4 macromolecules. What is an enzyme? Is it a carb? This is from unit 2 of semester 1. Carb, fat, protein, nucleic acid. What is an enzyme? An enzyme is an example of you're right, it does work on breaking down things. Which one of those 4 macromolecules in our body is it? Enzyme is a what? Is it a carb? Is it a protein? Is it a lipid? Is it a nucleic acid? Yes, it is a protein. All right. proteins. Remember, we talked all the way back then about why those are so important. Well, guess what? It is January, guys, and we are still talking about proteins. All right, proteins are so important. And this enzyme, this protein lactase, is super important because our bodies cannot break down lactose if we do not have this enzyme. So if you are lactose intolerant, you are not producing a protein that others of us are. All right. So that is just like solving mysteries for you right now. Okay. And E. coli, prokaryotic that live in your large intestines, they try to break down our lactose. All right. How do they know when they get it? Well, when something comes in, when it's present in your intestines, is when that enzyme is going to get to work. All right. So, what do I want you to remember about the lac operon today? Well, I want you to remember that it's associated with lactase and lactose, which is really easy to remember because what might be a good indicator? Use your pointers to show me why we might remember that the lac operon is responsible for breaking down lactose. Yeah, the LAC, right? Isn't that helpful? The LAC in the lac operon is going to help us remember that it is associated with our lactase and our lactose. So let's look at this diagram that I made, okay? Some diagrams are just not good, and I was like, I like this one. All right, so here's our gene expression. A gene is a piece of DNA that controls one of the hundreds of thousands of characteristics we've had. That's from a past unit, right? That is controlled by transcription, translation, and the protein that is produced. All right. In the case of the lac operon, that would be lactase. All right. This is the protein, that's the enzyme that we were looking at that is expressed. How do we stop the expression of a gene? So let's say we have a gene that we need to turn off. Something isn't present and we don't need that little Pac-Man in there to keep going around and breaking things down. Any ideas? How might our DNA stop it? Ooh, Dustin's been reading ahead. He's got some vocab words he's pulling out. Pip says the same thing. Well, let's look. Let's pick. So the lac operon regulates gene expression based on the cell's environment. So here, this circular tube in this example is our DNA, okay? We have a segment of our DNA that we talked about. So this is one of the chapters. We're pulling out the major Lego manual, and we're flipping just to the page of our project, just of our lac operon, or our lactose project, right? This is kind of what it looks like. And I kind of love that it's colors because Legos are colors too, right? So this first piece is what we call a promoter. Everybody type promoter. I also love these words because they make sense.
Promoter. In other words, a word from our sponsor, Raid Shadow Legends. I'm just kidding.
Yeah, right? So when you promote something, you're getting ready to express it, right? You're announcing it. You're saying, here comes the lac operon, right? Right? So this is kind of like the introduction to what needs to happen in the sequence. So what sometimes is the base for some of your Lego projects? I'm thinking of one piece that would really represent the promoter. Yes. You guys know what I'm talking about with the base plate? Pip said it too. It's the big green plate typically. The big green plate that you build all the pieces on. Yeah, that's what I would think the promoter is. They can be gray, they can be, yeah, if you get the different kits, they're different colors. So the promoter is like that big Lego plate, blue, green, whatever it is. All right. And then we continue reading. And I love a different color, right? Next step is what we call our operator. And I kind of like to think of this piece of DNA as the one that's saying, all right, the code is coming. Like kind of like the MC that's like going to be calling out the different pieces. Yeah, you're absolutely right, Jesse. I love that. Like this piece of DNA is like the motherboard of your body. 100%. 100%. All right. And then just like our Legos, instruction tells us each piece of what we're going to need. This is going to tell us our different organelles in our cell what to do, where to go, what to produce, how much of it to produce, whatever its role is. Okay. And then we're going to get to this third part of it. And that is going to be responsible for who knows what. What do you think the last part of that might be for? What's at the end of a lot of our traditional storybooks? If I was reading like Little Red Riding Hood, sometimes there's a page at the end. And it says, yes, Kaden, it says the end. Or it says like they lived happily ever after, right? It tells you that it is the end. Like that is that's the final, that's where I want you to stop, right? All right, so this is what we kind of look like as we go through and read it. Right, we have our segment of DNA, and we have our RNA polymerase. It kind of looks like a Ziploc baggie. Does that make sense? It's going to go in and copy that. Yes. Doesn't that make so much sense? I love it. So I want to show you this in action. Let me check our time. We have time. Let me check and pull up a good video of this because it's super easy to be like, oh, this is how it works. Would somebody, while I pull it up, let's do our own drawing and interpretation of it, okay? Our Lego book is going to tell our lac operon where to go, what to do, and how to do it. Here we go. Here's our whiteboard. Let's go to a new page. I want to put gene expression at the top, right? Because a gene is a piece of our DNA. We're reading our DNA. We're reading that instruction set for any given thing, particularly the lac operon, okay? Can you guys work collectively together? I want you to put that little drawing together while I pull this video up. Oh, my internet's being slow. It's doing the spinny thing today. Let's see, how are we doing? Oh, we're struggling. All right, well, somebody draw me a piece of DNA. Somebody draw me a piece of DNA. You guys can do that. Perfect. All right. There's our DNA. Now somebody highlight a segment of that DNA to separate out the gene. You guys are going to be excited about this video. Wait. All right. There we go. There's a segment. Good. And what's going to be at the very first part of that code, of that gene code? Yeah, what do we call the very first part though? Operon, promoter, the end. Trying to give it to you. I know this is a really complex. This is a complex lesson. There we go. There we go. Our promoter. Remember, we're saying, here we go. Indicating the start. All right, let's go to this video. I know you're going to like it. And let's take a peek. Are you ready? And I want you to, please say my internet can keep up with it today. I'm going to turn me off for a minute so I'm not fighting with it. Not giving it a reason to not. I am not seeing it. Do you see them? I am not seeing them. Okay. Some of us are seeing it. Some of us are not. Maybe I'm going to also put it in chat so I don't disrupt those of you who are seeing it. Okay. For those of you that are watching, watch. For those of you who are, I just put the link in chat. And some of us see it and it's not playing. I could also desktop share. Want me to do that? All right, here. Let me get it for you. Here we go. Are we seeing it now? Let me get us past the ads. All right, here we go. Can you hear? Perfect.
Have you ever thought about what a disaster it would be if the cells in your eyes started producing the same hydrochloric acid that is made by your stomach cells? Your stomach cells produce hydrochloric acid to help break down food, but you definitely don't want that in your eyes. Thank goodness that doesn't happen. But it's surprising because both your eye cells and your stomach cells contain all of your DNA. Your DNA, all of it, is found in your body cells. But see, the portions that are used need to be regulated somehow. Otherwise, we could end up with something ridiculous like eye cells producing hydrochloric acid. And that wouldn't just be a waste of resources, that would actually be very difficult to explain to your friends. You want genes to be regulated, controlled. Remember that genes are made up of DNA. DNA is used to give instructions for the production of proteins in the process of protein synthesis. But an important concept is that there needs to be a method of determining which genes will be turned on and which genes will be turned off. This is called gene regulation. And there are many ways that genes are regulated. In your human body cells, you can have proteins that can bind to certain gene regions to increase the rate of transcription for the transcription enzyme RNA polymerase. Or you can have proteins decrease transcription to the point that it may not be transcribed at all. This is a form of gene regulation. Your eye cells don't use the portion of DNA that codes for making hydrochloric acid like your stomach cells do, because there is regulation in your cells to determine which portions of the DNA is used. But we want to shift gears now to talk about a very interesting way of regulating genes that can be sometimes a little challenging to visualize. This particular method is really found in prokaryotes, a few eukaryote exceptions. It's called an operon. An operon is a fancy way of regulating genes, and it's usually made up of a few genes that can involve enzymes. Remember that enzymes are proteins with the ability to break down or build up the substances that they act on. Now let's talk about some key players in an operon so that we can see some gene regulation here. First, RNA polymerase. It's a builder enzyme actually, because RNA polymerase is an enzyme. In biology, a lot of enzymes end in that ASE. RNA polymerase is needed in order to start transcription. Remember that transcription and translation are steps in protein synthesis. Protein synthesis means to make proteins, enzymes in this case. The thing about RNA polymerase, though, it gets a little confusing for RNA polymerase without somewhere to bind. If you watched our DNA replication video, you learned about DNA polymerase and how it had to have a primer to know where to start. Well, RNA polymerase needs a promoter. A promoter is a sequence of DNA where the RNA polymerase can bind. So you would think, okay, you got your RNA polymerase, you attach it to a promoter, and boom, you make your mRNA, which eventually will be used to make a protein. But there's this other sequence of DNA called an operator. The operator is a part of the DNA where something called a repressor can bind. The repressor, if bound to the operator, blocks RNA polymerase. And poor RNA polymerase cannot move forward and no mRNA can be made. Therefore, no proteins. So let's take a look at our setup here. This is an example called a lac operon. Notice there's a promoter region of the DNA, the operator region of the DNA.
Okay. So it's just getting good. All right. The Amoeba sisters are just so good. So what we're looking at today, the prokaryote cells are what we're looking at today with our lac operon, right? So prokaryotic cells, are they cells that we are made out of? No, remember, prokaryotic cells are our simple cells, right? So we're going to look at how that works in simple cells. So we did something that is very similar to this last semester. I think sometimes that's what makes it so confusing, is because the processes of transcription and translation in eukaryotic cells, when we're going to make proteins, remember, we go to transcription, translation to our ribosome, we make our proteins, and they go wherever they need to go. That process is very similar to what we're talking about. So sometimes I think that's why it's like, woo. What I want to do today is, before we continue to go into the lac operon example, is I want to make sure that we get what the whole beginning chunk of that is. All right. Is that what we're looking at is how our genes are expressed in each one of our cells, whether it be a plant cell or a human cell or a piece of moss. Okay. And we talked last week about how that, pretend there's a wall in your house that's full of light switches, just hundreds of light switches, and that each one of those represent a way of turning off or on a piece of DNA that you need, right? So for each one of your cells, most of those switches are turned off. Just like our Lego manual of all possible projects. For our cells, you just need a couple pieces of those chapters in order for them to do their job. Just like they talked about, your stomach cells are going to use a whole bunch of different information than your eyeball cells are going to use. And that they can kind of forget that other information and focus on their job, just like we do, right? So we all go through high school and then we go get some education that makes us specifically skilled or trained at a job, right? So I went through high school and I went through college, and I took a bunch of classes that were not biology related to give me a rounded education. And then I took a bunch of way more biology classes than maybe someone that is going into writing, right? They aren't taking biology classes. They're talking about ways of expressing themselves through words. And, so they took a whole different path. So I kind of like to think about our cells' journey from the day they're born to the day that they're doing their job, to the same way we are. All right. We're going to watch the rest of that video tomorrow, but I want to end this lesson with you feeling like thumbs up about what a gene is, and what it like visually looks like to have a piece of information that you turn on and turn off. And we talked about that promoter makes sense, and some of that reading of the DNA makes sense based on what we've previously learned. But this is a big thing to unpack. Even the Amoeba sisters said that, right? So you're doing a great job for a Monday. I'm super, super duper proud of you. We did not get all these goals accomplished today, right? Because this is something that's going to take us a couple days to get through. But we did get through what? What is a lac operon? Do you feel like we accomplished that goal today? Show me. Good. Now we looked a little bit about when is a lac operon turned on, right? We talked about that promoter turning that being an indicator to turn that on for gene expression. Absolutely, Kai. If you think you're ready to take that quiz, you're welcome to take that today. Remember, you have multiple attempts on that, so that's an okay thing to do. That's just going to increase your learning. But again, you're welcome to take it again tomorrow after class, and we'll do this quick check tomorrow after class. That'll kind of preview those questions, right? And make you feel confident going into it. So we covered about half of our information today, but that's okay. I'd rather get through half of our information and feel good and decent and not overwhelmed than push through this tricky concept and have you guys do that. I'm going to put that link again in chat so that if you want to go look at that and watch that whole video on your own time, and I'll also put it in the announcement section of our classroom. All right, so that, yep, we're going to peel it away at an onion, one piece at a time. Absolutely. I'm super proud of you. Again, what are you going to do to take this to the next level? I'm here to teach it to you. You've got to take a step. So I want you to indicate what you're going to do today on your own. Are you going to start making some flashcards or maybe do a drawing? Are you going to go in and do the reading if you haven't done the reading yet? Are you going to watch our recording again? That would be a good thing to do. Or watch the Amoeba sister video that I sent. Okay? Wait, remember that this class doesn't stop with me teaching you that you've got to take it that extra piece of the way. Yes, Jesse, we've all got classes we need to run to, so feel free to head out. Thank you so much for being here, guys. I'm glad you made it back here this snowy Monday. We're going to tackle the rest of this tomorrow. You did a great job. Feel better, Aria. I'm so sorry. It's no fun being sick. Anybody else have questions? That vitamin C will help you.